This lesson is a continuation of our plant diversity unit. In the previous lesson, we talked about the evolutionary origins of terrestrial plants. We'll spend more time in lab talking about plant systematics and classification. And in this lesson, we'll do a survey of the plant kingdom, but the emphasis will be on plant reproductive strategies. One of the things I want you to take away while you're thinking about plant reproductive strategies is how does this go back to specific evolutionary adaptations that we see in each group as they are progressively adapted to life on land. I'll try to point that out as we go through the life cycles as well. You probably recall from the plant evolution lecture the bryophytes. The bryophytes are actually what we consider a quote-unquote informal group. So by that we mean that it's a group of plants that aren't necessarily related to each other evolutionarily. There's some dispute about how closely related the informal group, the bryophytes, are. But we lump them together as the bryophytes because they represent the earliest plants on land. So the bryophytes are the representative group of the seedless, non-vascular plants. They include the mosses, the liverworts, and the hornworts. I'll focus on the mosses as our representative group from this particular informal group. The bryophytes include the earliest plants on land. Most have a cuticle and stomatal pores, but the cuticle tends to be much thinner and not really as waxy as the cuticle we see in plants that are more recent in evolutionary history. All of the bryophytes lack vascular tissues, so while they do have some primitive cells that allow for the transport of water and sugar, they don't have the more complex tissues that we learned about earlier in the semester. So for these two reasons, and for reasons that we'll see when we look at their reproductive cycle, the bryophytes must remain in moist environments. So the bryophytes are dependent upon an aquatic environment to survive. And then if you go way back to a lecture where we talked about the characteristics of plants, one of the major characteristics of plants we talked about, and I said I would explain later, was this idea of an alternation of generations. So plants go through a multicellular haploid and multicellular diploid phase. The multicellular haploid phase is called the gametophyte generation. This will make more sense when we look at an actual life cycle. In most plant groups, the gametophyte or the haploid generation is the smaller, less predominant generation. In bryophytes, they're unusual because the haploid generation is the dominant or predominant generation in the plant's life cycle. Again, I'll explain more about this when we get into the nuts and bolts of the bryophyte life cycle. So I'm using the mosses as our representative bryophyte, and I just wanted to quickly point out, I talked about the dominant generation being the gametophyte generation. So if you happen to pay attention to mosses next time you're out and about and looking at the seedless plants, look to see if you see these little stalks with the little capsules that are attached to them. The little stalk with the capsule that's attached to it, that's not as frequently observed on the mosses. That's actually the sporophyte generation, or the diploid part of the moss life cycle. The lower to the ground grassy stuff that we see in moss, that's the gametophyte generation, or the haploid part of the moss life cycle. So some general characteristics of mosses. We say they're low-growing and carpet-like. There's so many different species of mosses. We're still discovering new moss species. We say they have a thallus, but no true leaves, and rhizoids, but no true roots. And we say this because they do not have true vascular tissue. So because they don't have true vascular tissue, we say they have a thallus and rhizoids, not true leaves, not true roots. They are dependent on water for reproduction, and I'll explain why when we look at their life cycle. And again, their dominant generation in this alternation of generations, their dominant generation is the haploid gametophyte generation. So we are going to look at a cyclical life cycle, but I actually like to first present the life cycle in kind of a linear form, just to kind of introduce you to the terms and get you a little bit more familiar with thinking about life cycles. I do this because often when students see a cyclical life cycle, they get really confused because they're not really that used to seeing information presented in that way. So to get you comfortable with looking at the life cycles in a cyclical format, I'm going to kind of introduce you to some of the terms and the sort of the flow of the life cycle for each of these groups. So in the mosses, we would start with a spore. The spore is a haploid spore, so that means it has half of the set of genetic information. We'll look at this again when we look at the cyclical life cycle. The spore will germinate into this structure we call a protonema. And the protonema will continue to divide mitotically to form a thallus and the rhizoids. And on top of this thallus, we will see the new gametophyte generation. So the new male and female sexual spores are produced here on the thallus. And we'll look at that in the next slide. So the male gametophyte in the moss is found here on top of the thallus. I'm circling it here in red on the left-hand side. And that structure is zoomed in over here. And I'm going to circle an individual antheridium right here. And here it is blown up. So you can see all these antheridia. And inside of this antheridium are thousands of sperm cells. The antheridia will bust open when the sperm are mature, and they will move through a film of water. So they're flagellated, and they have to have water to move through. So they'll move through a film of water, and they're chemotactically attracted to exudates from the structure that houses the egg. And we're going to look at that in the next slide. So this is the moss female gametophyte. I think it kind of looks like a sideways alligator head. That's just me though. And here's the egg. And it makes sense that our sperm and egg would be haploid, right? So they're produced by the gametophyte and they are haploid. We're talking about the gametophyte generation of the plant. And if you recall, we had the sperm cells that were housed in the male gametophyte, the antheridium. The sperm cells will bust open, the antheridia will bust open, and the sperm will be chemotactically attracted to the egg. So the sperm actually swims down, it moves through a film of water. There are exudates, so secretions from the archegonia that attract the sperm to the egg, and then the sperm and egg come together and they will produce a new sporophyte. So a haploid sperm plus a haploid egg will result in a diploid new sporophyte generation. Oh, and if I didn't already say this, the structure that we're looking at is called the archegonia. I remember these words because we see egg here, so archegonia, house of egg. And the male gametophyte we looked at was called the antheridia. And so I just remember the word anther. Remember we saw the pollen on the anthers? We know that's the male reproductive structures. So just to recap, we had a sperm from the male gametophyte, the Antheridium, and an egg housed in the female gametophyte, the Archegonium. They come together, the sperm swims to the egg. They come together to form a new diploid sporophyte. So now we're talking about what we call 2N, or diploid, versus N, or haploid. So we have the new zygote will continue to divide mitotically, and we'll see this stalk shoot up. This is the new sporophyte. That's the stalk. And then on top of this stalk, we see this capsule. And the capsule contains sporangia. And these sporangia will undergo meiosis to become the spores that produce the new gametophyte generation. So there's spores that are contained in these capsules that will produce the new thalli that will house the Antheridia and the Archegonia. And the spores, because meiosis has occurred here, these are haploid. And the last few slides in this slide are just a recap. Here is a close-up of that sporophyte capsule and the sporangia inside this capsule. They will undergo meiosis to make haploid spores that germinate into the new gametophyte generation. For each of these groups of plants, after I give you this introduction, I'm going to go through and show you an actual cyclical life cycle. So we'll recap again by looking at the life cycles. But again, as I told you in the beginning, kind of want to just go through each of the representative groups, and then we'll go back through the life cycles. All right, so now we've made it to the vascular plants. So first group we talked about were seedless, non-vascular. Now we're looking at the vascular plants. The vascular plants have true roots and leaves because they have more modern, complex vascular tissue, xylem and phloem. All of the rest of the plants have a less predominant gametophyte generation. So even the ferns, even though the ferns are still seedless, even the ferns have a less predominant gametophyte generation than the bryophytes do. And then, of course, they have a larger or more predominant sporophyte generation. So let's look at the representative groups for the seedless vascular plants. And so I call this the ferns and the fern allies. And that would include the division Psallophida, division Lycophida, division Sphenophyta, division Pterophida. You have looked at some of these already in the Common Plants of Wisconsin. So you had a club moss, you saw the horsetails, you saw some ferns in your common plants, and then hopefully you've seen some of these in your adventures that you've had as you've gone out looking for these. So we're going to look at the ferns as a representative group, and we'll briefly look at the fern life cycle. But again, just a reminder, we'll also go through the fern life cycle kind of in a cyclical format as well. So ferns aren't really important in terms of commercial importance. And of course, that's sort of a subjective thing to say, the commercial importance of a plant. But they're still globally important as houseplants. They make up important parts of various ecological systems. We still have about 11,000 species of varying sizes. But in our next slide, we'll see how incredibly significant the ferns and the fern allies were during especially the Carboniferous era. So the ferns and the fern allies dominated during the Carboniferous era, which was approximately 300 million years ago. In fact, some people call this the age of oxygen. So during the first part of the Carboniferous era, oxygen levels were around 30%, which is almost 50% higher than current oxygen levels. Think about that. Oxygen levels during the Carboniferous era were almost 50% higher than oxygen levels on planet Earth now. So this in itself is fascinating. You see this picture of the great big dragonfly here? We actually know that insects were much larger during the Carboniferous era, and for a long time the belief was that insects could be larger because oxygen was so readily available. So we kind of have this stereotypical like giant um, arthropods from the Carboniferous era, and that's what the fossils suggest as well. But interestingly, some more recent research suggests that perhaps the insects were so large because only the larvae that were really big would survive the high levels of oxygen. In fact, the oxygen at really high levels was toxic to smaller insect larvae. So there's still so much fascinating information that we're still learning about this period in time. Another important outcome of the Carboniferous era, you note these massive forests. So there's these massive swamp-like forests of the ferns and the fern allies. And so we're talking about trees as big or bigger than some of the largest gymnosperms and angiosperm trees that you've seen. So big, massive forests of ferns. Most of these species no longer exist. But these trees, after the oceans flooded these swamps, these trees actually became our major source of coal deposits and some other fossil fuels. So even though I said in my previous slide there's not a lot of modern day economic value in the ferns, they actually left behind some of the most important and valuable resources that we have available to us on the planet, particularly post-industrial revolution resources. So I'll try to find some articles for you on the Carboniferous era if you're interested, and just put them in your additional resources folder. These are just optional reads, but this is such a fascinating era. It's kind of something fun that you may want to explore on your own. So just a few fern characteristics. We call the big feathery leaves fronds, Fronds, fronds. And they are still leaves, but typically botanists will call fern leaves the frond. And although ferns have true leaves and true roots, their vascular tissue isn't exactly the same as the vascular tissue that we see in the angiosperms and the gymnosperms. So we could actually distinguish ferns by their vascular tissue. Most ferns will also have a rhizome, this underground stem that new ferns will shoot up from. So in ferns, if you were to say pull up some ferns, you'd see a few roots. Typically they're fairly shallow. The petiole and the blade of the frond. And although these have vascular tissue, they are still seedless plants. So they don't produce seeds, they don't produce fruit, they don't produce flowers. And they are dependent on water for reproduction. And I'll explain. Very, very similar life cycle, in fact, to the mosses. And we'll look at why they require water for reproduction in the upcoming slides. Like all plants, ferns undergo an alternation of generations, the multicellular haploid and the multicellular diploid parts of their life cycle. As I'm talking about ferns today, we'll start with this sporophyte generation stage that you may be familiar with seeing in the spring, called the fiddlehead stage or the crozier stage. You might be familiar with this because some people will actually go out and collect fiddleheads from some fern species. My dad did this. In fact, he would go collect bracken fern fiddleheads. And I guess he figured he had so many kids, he had 11 kids, he could experiment with us a little bit. We lived. They were okay. They didn't taste that great, but you can eat some fern fiddleheads. But anyway, I mention this because it might be something that you're familiar with. This fiddlehead will unfurl and it will mature into these large fronds. And here you can see at some point these big ferns will have new fiddleheads popping out throughout their growing season. And then you can look on the underside of a fern leaf and you'll see a bunch of sori. And sori remind me of that candy that you would get on pieces of paper. And there'd be those little tiny dots of candy, and you try to peel the candy off, and some paper would stick to it. So that's what sori look like. The next time you're out and about, maybe you're doing your plant scavenger hunt, look for some ferns. Look on the underside of the leaves of ferns, and you'll probably find some of the ferns that have these sori. So the sori in this example, maybe I have a better picture, I can't remember, but I'll point it out in this one. In this example, you see this little green circle? This little green circle is actually a protective layer called an indusium. Not all sori have that. In fact, ferns have all kinds of variations of their sori. We might have like a linear sorus. We might have just a circle with no indusium. They might be arranged and organized differently. That can actually be one way that we're characterizing the type of fern. Oh, and you also saw like the ferns like the cinnamon fern and the interrupted fern that actually have some specialized megaphylls that, in fact, rather than being leaves, they bear the sori. So it's not always just on the undersides of leaves. So then in addition to this indusium, you probably see this little black circle, and there's lots of little black circles. So these are sporangia, and they contain spores that, just like we saw in the mosses, the spores contained inside that sporangium will undergo meiosis to produce a haploid spore. That haploid spore will germinate to produce the new gametophyte generation. So really what we're looking at here is all the sporophyte, the diploid, or the 2N part of the fern life cycle. Okay, good. I had some better sori pictures here. So this is a sorus, just like I was explaining to you on the previous slide, but this is a nicer picture. This sorus has an indusium. Not all sori have the indusium, but you can see this kind of little protective cover on the fern, or on the sorus. And then you can see here the sporangia that I was talking about. And here's a blow up of a sporangium with the spores inside. See these little spores? This is after meiosis has occurred. So now these spores are haploid, or 1N. Before meiosis, they would be diploid, right? So now they're haploid spores, and they will mature into the new gametophyte generation of the fern. So again, going back to that idea of 2 generations in the plant's life cycle, the haploid generation and the diploid generation. So the gametophyte is the haploid. And the fern gametophyte is, between you and me, totally adorable. Look at this little thing. It is a heart shape. And on this heart shape, I'm going to move over to here. Oh, they're about the size, they vary in size, but they're like the size of your thumbprint, or maybe your pinky print. They can vary. But this is what most fern gametophytes look like. And then on this little heart-shaped thallus, we have the archegonium. Does that sound familiar? And the antheridium. So this is where the similarities to the moss life cycle are really apparent. So here we have this archegonium, and if you recall, archegonium meant house of the egg, and antheridium means house of sperm. So here the antheridium are circular here, and you can see the little sperm cells inside. So just like the mosses, the fern sperm cells are flagellated and they have to move through a film of water to get to the egg contained inside the archegonium. So the haploid sperm and the haploid egg will come together. Just like the mosses, the sperm is chemotactically attracted to the egg. So the sperm will swim to the egg and produce the new zygote, the 2N zygote, that will become the next sporophyte diploid generation. So here we just see some sperm cells actually swimming to the egg cell, and the new sporophyte will grow up off of the gametophyte. So this is that heart-shaped gametophyte right here, and right here is the new sporophyte that will grow into those crow's ears, the fiddleheads, that I showed you at the very beginning of our discussion of the fern life cycle. So we are moving up the evolutionary ladder here, right? We looked at the mosses and the other bryophytes, and then we looked at the ferns, and now we'll look at the gymnosperms. So the gymnosperms have seeds that are exposed on sporophylls. And sporophylls are a type of modified leaf that bears some type of a sporangium. We call these plants heterosporous, and we'll look at that in an upcoming slide. That means that unlike the other plants, these plants are actually producing two different types of spores, a megaspore and a microspore. And we'll talk more about that when we look at the life cycle more closely. This group includes 4 divisions: the Pinophyta, the Ginkgophyta, the Cycadophyta, and the Gnetophyta. Mostly, there's not a lot of species left in these divisions, and so I primarily use the Pinophyta as our representative group because it by far makes up the majority of the species that remain in the gymnosperms. So for this group, the gymnosperms, we use the Pinophyta as our representative group. They are the dominant plants as trees in the northern hemisphere. There is a lot of diversity of species and genera. You've learned some of these in your Common Plants of Wisconsin. They're also fascinating because they include some of our oldest known organisms. So there are some gymnosperms that are over 4,500 years old. So they have a fascinating history. And in the upcoming slides, we'll talk about their unique life cycles. The Pinophyta is a fascinating group with a fascinating evolutionary history. Although all of the currently living species of Pinophyta are woody plants, if we look at the evolutionary history of the Pinophyta, there's actually a number of species that were herbaceous and not woody. What's surprising is that the Pinophyta that survive today almost all have characteristics that allow them to survive in very cold and snowy climates. For example, the shape of most Pinophyta allows for snow to kind of slide off of it. They also often will have biochemical properties that allow them to survive very cold winters. Another characteristic of most Pinophyta is their needle-like leaves. So the needle-like leaf actually reduces the surface area, and the leaf typically has a very thick cuticle with sunken stomatal pores that helps them to keep from drying out and freezing in really cold climates. So I also want to point out, because we didn't get to do a whole lot with pine identification, when we look at a dichotomous key for pine, one of the first things they'll ask you is, how are the needles organized? And in a lot of the Pinophyta, the needles are clustered together. And in that cluster, we refer to it as a fascicle. So a fascicle is when we look at one kind of region and we see multiple needles coming off from one point. So look at a white pine, and we'll note that there's 5 needles clustered in a fascicle on a white pine. So as I mentioned previously, the Pinophyta have many adaptations to harsh conditions, cold and dry. And actually there's a large part of Florida that's very sandy, and although it's really hot, the pines tend to do really well in this sandy area because they're also adapted to really dry habitats. So there are some pines that are more of a southern species that we'll find in these drier habitats. And this is just a nice cross section of a needle, and it's showing some of those characteristics that I mentioned, the sunken stomatal pore and the thick cuticle layer that allows for, remember we talked about the leaf-air interface and the importance of kind of that boundary layer to prevent water loss in plants that are trying to restrict water loss if they're in dry regions. So the pine needle is really extremely well adapted to dry climates for this reason. So I mentioned earlier that the pine life cycle is heterosporous. That means it's producing 2 different types of spores. And it produces these 2 different types of spores on 2 different types of cones. So there is the staminate cone, or the pollen cone. And on the staminate cone we have microsporangia that produce microspores that essentially mature into pollen. And the pine can make tons of pollen. If you've ever parked underneath pine trees in the spring, or if you looked in mud puddles in the spring, you've probably seen just the millions and millions and millions of pollen grains that are in the air and all around the pines. So in this slide we see the male pollen cone, and here is a close-up of that. And you can see the microspores inside the male cone here, and then those microspores mature into these pollen grains. The microspores are haploid, and they have originated from a cell called the microspore mother cell, or a synonym for that would be a microsporocyte. So the microspore mother cell, or the microsporocyte, whichever you prefer to use, is a diploid cell that undergoes meiosis to produce the haploid microspores. And then the haploid microspores will develop into this pollen grain. And the pollen grain has here in the center something called a generative cell. That will actually divide into the sperm cells that we see in the pollen grain. There's also what we call the wings, or the bladder, of the pollen grain. This got 2 functions. One, the wings of that pollen grain help it to move through the air. So this is a huge evolutionary adaptation, right? This sperm is moving through the air, not dependent upon water. And in addition, this pollen grain at some point will produce a pollen tube that the sperm cells can move through. Now it won't produce that until it actually reaches the female cone, but it's a huge adaptation. So these are some big advantages that we see in the gymnosperms. So recall we said the pines were heterosporous. That meant they produced 2 types of spores. And so we talked about the male spores, and now we'll talk about the female. So these are produced on the ovulate cones, or the seed cones. These are the cones that you're probably most familiar with thinking about when you think about pine cones. And inside the ovules, there are megasporangia, and I should just take a little aside here. Some people will use the term megasporangium as synonymous with ovules. In fact, I personally think the term ovule is a lot less confusing for people, but very technically speaking, an ovule is a megasporangium with integument surrounding it. But anyways, we'll see this megasporangium producing megaspores, and we'll look at this more closely. We'll look at this process more closely in an upcoming slide. And importantly, we also see this entire seed covered by integument. Remember we said an ovule is a megasporangium covered by an integument. And this is important because that becomes a seed coat. So recall, one of the huge adaptations that we see in the pines is that in addition to no longer having a dependency to move the sperm through water, we actually see seeds. So the plants before this were seedless. And so now we're looking at the formation of seeds. And here we have a cross section of the female cone where we see the ovule. And the term ovule, I think I mentioned previously, is basically synonymous with the term megasporangium. And inside this megasporangium, so if this was our ovule, excuse me, what I'm highlighting here before meiosis occurs would be megasporangium, and then including the integument would be ovule. Well, inside the megasporangium, there is the cell called the megasporocyte, or the megaspore mother cell. That cell is going to undergo meiosis to produce 4 megaspores. 3 of those megaspores will die, and 1 will become the megagametophyte. And the megagametophyte will actually contain archegonia that have the egg, that contain the egg. There's 2 or 3 typically in the Pinophyta, 2 or 3 eggs in the female gametophyte. After fertilization occurs, so after the pollen grains reach the megagametophyte and fertilize the eggs, this megagametophyte becomes nutritive tissue. So different pine species do different things with this megagametophyte. Some pine species will actually shuttle additional nutrients to the megagametophyte, and others just have this megagametophyte basically be nutritive tissue for the developing embryo. So key points here, we have the megagametophyte being our gametophyte generation, right? We talked about every plant having the alternation of generations. So in the male spores, we'd see the pollen grain being our male gametophyte, the male haploid generation, the male multicellular haploid generation. And here we have the female gametophyte, the megagametophyte. It's haploid, and it contains the archegonia with the eggs, and it is the haploid generation of the pine life cycle. And then of course the new embryo is diploid, and it will produce the new sporophyte generation. And now we're moving on to the most recent plants in evolutionary history, the angiosperms. So the word gymnosperms means naked seed. The word angiosperm means covered seed, or seeds in a vessel. And in the angiosperms, that vessel is the fruit, a ripened ovary. So in the angiosperms, we have incredible diversity. I have the number 250 because you see that reported a lot, but more recent estimates bring this up to closer to 500,000 species of angiosperms. Hundreds of families. This is what we've spent most of our time on. So there's great diversity, great economic value in the angiosperms. And there's There's two major classes of angiosperms, which we've talked a bit about, the dicots and the monocots. And so earlier in the semester, we contrasted the monocots with the dicots. We won't spend a ton of time on that. In some of our upcoming labs, the lab that you're going to go on, do your plant family activity, we might spend a little bit more time thinking about these major classes of the angiosperms. So it's pretty easy to distinguish the angiosperms from the other groups because the angiosperms have not only the seeds and all of the other adaptations that we saw in the previous groups, but they have flowers and fruits. For the rest of the slideshow, I'm going to go through the plant life cycles again. I will actually be looking at the cyclical life cycle in the slide. And we should spend some time comparing and contrasting the reproduction in each of those four representative groups and really pointing out the key evolutionary distinctions or adaptations that we see in each of these representative groups that we can identify from the plant life cycle. So with all plant life cycles, all of them, regardless of where they are in terms of their evolutionary history, remember, we could have a generic life cycle where we're looking at the haploid part of the life, here in the light blue, and then the diploid part of the life cycle, here in the red. Now, in the mosses and the ferns, particularly in the mosses, we see that the diploid, or the sporophyte, part of the life cycle is less predominant, but regardless, in all the plants, we could set up a generic life cycle, take away all the details here, set up a generic life cycle where we talk about the haploid and the diploid portions of the life cycle. So let's look at a cyclical life cycle for moss, and let's start here with these spores. You probably recall that these spores are haploid because here in this capsule, there's sporangium that underwent meiosis to produce the haploid spores. And the haploid spores germinated to produce a male gametophyte and a female gametophyte. And note that little N right here. That N tells us there's just one set of chromosomes, so this is haploid. And the male gametophyte bears sperm in the antheridia. And you can see in this little cartoon, the sperm cells move through a film of water. In this case, they're saying raindrops. They'll move through a film of water, chemotactically attracted to the egg. And the eggs are held on the archegonia. So here's a single archegonium that contains an egg. Sperm and egg come together to produce a diploid zygote. Mitotic cell division continues. So here we just see this continuing development of that zygote to an embryo, to a young sporophyte, to a mature sporophyte, and the mature sporophyte has this stalk with the capsule. The capsule contains sporangia that will undergo meiosis to make the cycle just continue. So key takeaways here: dependency on water, reduced sporophyte portion of the life cycle. So sporophyte is diploid, N plus N, 2N. And in the moss life cycle, we see that as reduced, not as significant. And the fern life cycle is fairly similar to the moss life cycle. Let's start here with the sporangium. So this is fairly similar to what we saw in the moss life cycle, but rather than a sporangium, I called it a capsule. Right, so the capsule or the sporangium contains spores. It will undergo meiosis. The spores undergo meiosis to produce haploid spores. That's what we see being released over here. These haploid spores are dispersed. So haploid N. That spore will germinate and produce a new gametophyte, this adorable little heart-shaped critter. And on this gametophyte, we have the archegonia and the antheridia. And the antheridia obviously house the sperm, and the archegonia house the egg. The antheridia, just like we saw in the mosses, have to move through a film of water. Sperm and egg come together to produce a diploid, 2N, zygote. And the new sporophyte, the new diploid sporophyte, will grow up off of the gametophyte. And then if you remember, we have these megaphils, or fronds. And often on the ferns, the sori that contain the sporangia are contained on the back side of the fern leaf. And then, once we get to the sporangium here, we're right back where we started. So the sporangium contains spores that undergo meiosis to produce the new gametophyte generation, and then sperm and egg come together to produce our sporophyte or diploid generation here in ferns. So now we're looking at a pine life cycle or gymnosperm life cycle. And it's a little bit different than the life cycles that we looked at previously, because now we're talking about a heterosporous type of plant. So it has two types of spores, but it's actually pretty much the same thing. We still have that generic haploid or gametophyte part of the life cycle and the diploid or sporophyte part of the life cycle. So we'll start here at number 1, and we're starting by looking at an ovulate cone, the female cone, and the male cone. And remember, these are actually, another term for them is sporophyll, and a sporophyll is just a modified leaf that contains a type of sporangia. So this is actually called the microsporophyll, if you want to get really technical, and this is the megasporophyll. So micro is typically used to refer to the male cells, and mega for the female cells. So the microsporophyll contains a microspore mother cell, or a microsporangium, that will undergo meiosis to produce microspores. Those microspores will develop into a pollen grain. So the pollen grain is basically the male gametophyte of the pine. And here's our ovulate cone. So what we're looking at with our ovulate cone, here's our ovule. And we talked about the ovule essentially being the same thing as the megasporangium, except the ovule also contains the integuments. So inside the megasporangium, there's a cell called the megaspore mother cell, or another word for that is megasporocyte. This megasporocyte or megaspore mother cell undergoes meiosis, and we see that here, number 5. So through this process of meiosis, the megaspore mother cell will produce 4 haploid cells, and usually 3 of those cells die, leaving just 1 megaspore. The megaspore will become the female gametophyte, and it will contain archegonia that hold the eggs. Usually in this female gametophyte, there are several archegonia, so there's several eggs, and although most often just one gets fertilized. So here we have our female gametophyte with several archegonia, and the archegonia contain eggs. And remember, this is our haploid gametophyte generation here, the female gametophyte generation right there. And we see the sperm and egg coming together, and then we have a new diploid sporophyte. The gametophyte serves as a nutritive source for the developing embryo, and eventually we'll get to a seedling and the mature sporophyte, or the big tree, and we start all over again. So it's a little bit more confusing, and I do think reading it in a textbook is helpful. Now, I've provided you with a chapter on plant evolution that goes through the life cycles. It also goes through plant evolution. So please read that. It's under optional resources. But you can also go to your individual chapters in your textbook. So you could read the gymnosperm chapter, you could read the angiosperm chapter, you could read the moss chapter. Really, the reason that I provided you with one unified chapter is because I think it's better for students to look at these as a whole, because one of the important takeaways is kind of a compare and contrast between each of the groups when we're talking about evolutionary adaptations in their life cycles. And remember, I know it gets boring to listen to me. You can stop and come back at any point. So if you get sick of listening to me, turn me off, come back and continue. I included this slide because sometimes you may go on and take another plant science class or another biology class, and some classes get really kind of nitpicky about which tissue is diploid and which tissue is haploid. And so if we're looking at this seed and we're wondering, like, what is the source of everything, where did each of these points come from, this seed coat, or that integument that we saw from a few previous slides, that's actually diploid, and that's sporophyte tissue. The gametophytic tissue inside, mostly what we have inside of this is haploid, because if you remember, this was all from the megaspore. Remember the megaspore mother cell that underwent meiosis, and we had the megaspore that matured into the female gametophyte. And so this is, for the most part, gametophytic tissue that's haploid. And then inside of this, though, remember we had the archegonia that had the egg. So inside of that we'll have, after fertilization, the diploid embryo. We could break this up even further, but mostly I point this out because, you know, in the future you may have a course where they get a little bit more particular about what's haploid, what's diploid tissue, and what was the source of it. So now we've come to angiosperm sexual reproduction. And angiosperm sexual reproduction is more complex than the life cycles that we've seen previously. One reason it's more complex is because while it is still heterosporous, we actually see a unique feature called double fertilization happening in angiosperms. So I'll explain that. I actually break this slide down into a series of slides so we can look at some of the details. One major point, if you do not know your parts of the flower, now is really the time for you to stop and refresh yourself on the parts of the flower. So feel free to just turn me off, go back and refresh yourself on parts of the flower. But let's start here. So we're going to look at the ovule of the angiosperm, and just like we saw with our other plants, we have what we saw with the gymnosperms, we have a megaspore mother cell, or a megasporocyte. The megaspore mother cell will undergo meiosis, to produce 4 megaspores. 3 of these die. 3 of the megaspores die, leaving 1 megaspore. The megaspore will divide mitotically a series of times. So we'll have this female gametophyte here with 8 nuclei, and the whole thing is haploid. So in an upcoming slide, I'm going to break this down for you, but just to recap what I just talked about, we have in our ovule, over here, a megaspore mother cell, a megasporocyte, that will undergo meiosis to produce 4 haploid megaspores. 3 of those megaspores die, leaving 1 megaspore. It's haploid. The megaspore will divide mitotically to produce the female gametophyte with 8 haploid nuclei. And we will come back to that in a second, but I just wanted to kind of give you a little bit of an introduction to this. So here we can see the male gametophyte, the pollen grains, right? And we'll look at that more closely in an upcoming slide. And you can see the male gametophyte landing on the stigma. It will literally eat through the style, produces this pollen tube. It's eating through the style. And there are 2 sperm cells contained in the pollen grain. 1 sperm cell will fertilize the egg, and the other sperm cell will fertilize some of those nuclei, some of the other nuclei that were contained in our female gametophyte. And we'll look at that all more closely. So let's look at the male gametophyte just for a minute. The male gametophyte, as you know, originates from a microspore mother cell or a microsporocyte. Sometimes you'll see pollen mother cell. So this is diploid, 2N. The microspore mother cell will undergo meiosis to produce microspores, and the microspores will mature into separate pollen grains. Pollen grains are really interesting. They will typically have a really thick outer wall that helps to keep them from drying out. Often there'll be these deep pits. The pits can help the stigma recognize the source of the pollen. There is also a generative nucleus that will form 2 sperm cells, and a pollen tube nucleus that will form that pollen tube that I showed you in the previous slide. So in this slide, I really want to focus on the formation of the embryo sac. We talked about pollen in the previous slide, so you can use this kind of a refresher if you like, but what I really want to focus on is the female gametophyte. And so we talked about this a little bit. We said that in the ovule, there is a megaspore mother cell, or a megasporocyte. That would be represented by this cell here. This will undergo meiosis to produce 4 haploid, 1N or N, megaspores. 3 of these will die, as indicated by my red scribble. 3 of those die, and then this remaining megaspore will divide mitotically a series of times until we have 8 nuclei. And these 8 nuclei will be organized like this. We have up here at the top 3 nuclei that are called antipodals, 2 nuclei in the center that are called polar nuclei, the egg here, and cells here called, or nuclei here called synergids. So let's talk about the function of each of these nuclei in the mature megagametophyte, or the female gametophyte, of the angiosperm. So the antipodals, for a long time, the antipodals were believed to be non-functional or vestigial. So they lost their function somewhere along the evolutionary chain. But in fact, we have learned that the antipodals could function in some way in terms of shuttling nutrients from outside. So once this is a seed and it's attached to some kind of a wall inside the ovary, the antipodals could serve as sort of facilitators of moving nutrients from outside to inside to serve the developing embryo. The polar nuclei will actually become a nutrition source for the new embryo, and we'll see how that happens in just a second. And the synergids here that surround the egg, the synergids basically act like, those people that help airplanes land and they have those flashing lights and they, direct airplanes on the runway? The synergids kind of act like that, but for sperm cells. So the synergids direct the sperm cell that's coming from the pollen. Remember the tube nucleus that's moving down the style, moving towards the egg. These synergids direct that pollen to the egg. So this is just another cartoon of the same thing I was just looking at. Here we have our female gametophyte contained inside the ovule, contained inside the ovary, right? And then after fertilization occurs, the ovary will actually, we use the word ripen, so it will mature into some kind of a fruit. I will put for you, I have a little video lecture on fruit types. It is optional, but I'll stick it in the additional resources. It's a voiceover, completely additional resources if you're interested. It is interesting, but it's up to you. I won't have it as exam material for you. Just a little recap here, everything I already talked about. And then this is just another recap of the mature female gametophyte, the antipodal cells up here at the top, polar nuclei in the center, the egg cell, and the synergids. And remember, at this point, everything here inside this gametophyte is haploid. So this represents our haploid portion of the angiosperm life cycle. This and the pollen grain would represent the gametophyte generation. And again, remember, plants go through that alternation of generations. They spend part of their life as multicellular haploid, and they spend part of their life as multicellular diploid. So let's say that a pollen grain from the right species lands on a receptive stigma. What is going to happen next? So we know that pollen grain has a generative cell and a tube nucleus cell. The tube nucleus will literally begin to eat away the style and will form a tube down the style heading towards the egg. Now there could be multiple pollen grains. Obviously sometimes we see that, and so there might be all kinds of, pollen tubes forming down this style. But really it's going to depend on whichever one reaches the egg first. So in this cartoon, the purple represents the pollen tube that has moved down the style, and now it's reached the ovule, and it has almost reached our egg. And we see the tube nucleus, and then we also see this generative cell. And this generative cell is actually going to divide into 2 sperm cells. So this is why we call it a double fertilization. So after the tube nucleus reaches the synergids, we'll see this generative cell divide into two sperm cells. And this picture just shows, over here I'm circling it in red, the generative cell dividing into two sperm cells. And so we can see this purple tube here, the pollen tube. One of the sperm cells is going to fertilize the egg. And then we'll get a diploid zygote. That's probably not a shocker for you, right? Probably what you expected. But in angiosperms, we have a double fertilization. And the other sperm cell fertilizes the polar nuclei. So both of these both of these nuclei are fertilized by this single sperm cell. So we get what we call a triploid, or a 3N endosperm. So the triploid endosperm is actually more nutrient dense than the haploid gametophytic tissue that we saw in the gymnosperm reproduction. So this is kind of an advantage for the angiosperms, this double fertilization, the 3N embryo. So we get two sperm cells. One will fertilize our egg, and the other fertilizes our endosperm. So at the end of all this, students will often ask me, why do we learn the life cycles, and why do we spend so much time on some of the details of the life cycles? And there's several answers to this. One, we can look at the life cycles and we can look at many aspects or compare and contrast aspects of the life cycle and see how new adaptations have occurred along this kind of evolutionary pathway. So major trend that I talked about in each of these life cycles is a decreased dependency on water and an increased protection of the embryo. But there's other interesting things that we can note from these life cycles. For example, when we compare the gymnosperm to the angiosperm life cycle, a big difference is the haploid endosperm, or the haploid gametophytic tissue that serves as nutritive tissue for the embryo, versus the triploid endosperm that results from double fertilization in the angiosperms. So the simple explanation of the advantage of a triploid endosperm is that, remember we talked about polyploidy, having multiple sets of chromosomes, will often lead to bigger tissues, more nutrient-dense tissues. Well, it makes sense that that's a big reason why the triploid endosperm is an advantage. But increasingly, that question's being asked, and there's some very, very interesting findings. For one, not all angiosperms have a triploid endosperm. Some angiosperms have other polyploidy levels of endosperms. Some angiosperms have diploid endosperm. And some angiosperms, I actually think it's water lily, they actually, rather than having a double fertilization like the way I described, they'll actually make little embryonic twins. So there's all kinds of unique strategies that are going on, and there's some interesting questions being asked, not just about, well, how does this serve the embryo, but also about the parental control over the developing embryo. So some recent literature is coming out suggesting that one reason there is kind of an evolutionary selection for the triploid endosperm has to do with the maternal to paternal ratio of the genetic information. So if you think of a triploid endosperm, that's kind of a 2-to-1 ratio of maternal to paternal genetic information. And they think, the researchers that are studying this, think that that has influenced the offspring in a evolutionarily predictable pattern. Anyway, I don't want to ramble too much. My point is that when we look at these types of differences in the life cycle, not only can we see major trends, but we can ask fascinating questions about plant evolutionary history and also about modern applications of that this might have in plant physiology and plant breeding and all kinds of things. So there is a purpose for this, and I think if you're struggling with life cycles, go back and just think about a generic life cycle. Remind yourself of the idea of the alternation of generations, how plants will go through the multicellular haploid phase, a multicellular diploid phase, and make sure that you can identify in each of the life cycles what is the haploid phase, what is the diploid phase. Start with that. All right, have a great day.